which covering mathematical and numerical modeling with applications to fluid mechanics, geophysics, medicine, and sports performance. So it gives me great pleasure to welcome him virtually to Glasgow to give the 2021 Sneddon Lecture as part of our distinguished lecture series. So thank you very much for coming.
Thank you very much for your very kind presentation and for inviting me. It's a big honor for me to be here today. So I will try to share my presentation first of all. I hope that it works finally. Now you should be able to see the title. Can you please confirm that you see the title?
Yes, that's fine.
Okay, very good. Thank you very much. So this is the title of the talk, A Bit of Math. And by that I mean using mathematics to try to simulate the beating of the heart. So it's about mathematical modeling of the heart. And let me first of all start by acknowledging the people who have been working on this project, and they include people from the EPFL in Lausanne, where this has started, this research has started, and mostly in Milano, Polytechnic Milano, where this project is going on now. So I will start with, for those who are completely unaware of the way our heart functions, by just a little bit of heart physiology. Heart is an organ which is made of 4 chambers. We have 2 atria in the upper part and 2 ventricles in the lower part. So the venous blood is brought to the heart through the vena cava, superior and inferior, and it enters the right atrium. Then it passes through this valve into the right ventricle, and then through this pulmonary valve, it enters this pulmonary artery. It goes to the lungs, where it releases the CO2 and other toxic substances, and it intakes oxygen, and then it returns to the heart. The oxygenated blood returns to the heart through 4 veins, which are called pulmonary veins. It enters the left atrium, and through this mitral valve, it enters the left ventricle, which is, let's say, the most important chamber, because it's the one that when it does contract, it ejects blood into the ascending aorta and through the aorta to the rest of the circulation. This is very quickly the way the heart functions as a pump, as a mechanical pump. But how is it activated? When does this pump take the energy? Well, the origin is due to the activation of this very point, which is called the sinoatrial node. Is in the upper part of the right atrium, and there is a spark of electricity that takes place from here. It propagates first to the upper part of the heart through the 2 atria, and then it reaches this node, which is a kind of gate. And this node is the atrioventricular node, sitting in the upper part of the ventricle, and through this and through the Purkinje fibers that you see here on the right, it reaches the apex and then all the cells of the myocardium. Now, this is the electrical component, and is actually the one which is behind the contraction and the relaxation of the heart. So to start with something which is familiar to the medical doctors, let me start with the ECG. The ECG is the electrocardiogram, and it looks more or less like this. It's a sequence of little waves, of wavelets, that in the, say, healthy case, resembles those that you see here. Now, as you know, this is captured by the ECG leads that stand on the chest of an individual. And if you go and isolate a single heartbeat, so 1 second, roughly speaking, of a physical time, you see the way this wavelet looks like. You have first the P wave, then you have what is called the QRS complex, and this is followed by the T wave, and then it is repeated over and over and over, right? This is seen at the level of, as I was saying before, of the chest. Now, when you go to the level of the cell, what you see is what is called the action potential. The action potential is the way you see the depolarization followed by a plateau and a repolarization. And again, this takes place over a single heartbeat. So in a time span of the order of 1 second. Now, this is what doctors recognize in terms of electrical behavior, but then you can associate this to any specific phase of the electrical activation. So you see the PU QRST waves here, and you see that component which is actually activated. We start at the very beginning from the resting configuration. Then you have here the sinusoidal node, the first activation, the first stimulus. It propagates first to the atria, and then you get the P wave that is activated. Then when you have the complete atria, you are between the P and the R wave. Then this electrical signal propagates through the Purkinje fibers, and now you have the R component of the QRS complex. Now the two ventricles are reached by the electricity, and then here you see the transition between the R and the T wave, and finally here you have the T wave when the complete activation has taken place at the ventricle level. So these are the different phases of the electrical activation, but then you can put them in correspondence with the mechanical behavior of the heart. You see again the PQRST complex here. Now again you go from 0 to 0.8. You know the frequency is not the same for everybody, but typically it's between 0.8 seconds and 1 second. So over this time lag, you see that you have different curves that are again appreciated by the doctors, and they refer to the ventricular volume, the way the volume of the ventricle changes over the time, the ventricular pressure, the atrial pressure, the aortic pressure. So it is this very picture which combines the essential features of the electrical activation of the heart and its say mechanical behavior through the opening and closing on the valves and the contraction and the relaxation of the ventricles that somehow characterize a heartbeat. Again, here is kind of atlas picture that is very well known to doctors. But what is the role of a mathematician? What is our aim? What would you like to add to this type of knowledge? Well, we like to end up with a model, with a mathematical model in terms of equations of the heart function. And before discussing this mathematical model, we have first of all to recognize that in order to simulate a heartbeat, we need first of all to start from a patient, hopefully from a real patient. So from medical images. And from medical images, trying to reconstruct shapes, forms, and geometries, right? Like the one that you see here on the left, for instance. So we need first of all to reconstruct the shape of a real heart. And this is not a simple task. It needs a lot of geometry. I will not have the time to discuss about that, but this is a kind of pre-processing. So once you have the shape, the second step is to generate a grid. And for those of you who are completely unaware of numerical approximation, a grid is necessary to describe a shape in terms of elementary shapes, like triangles of quadrilaterals in 2D, or tetrahedra or ixahedra in 3D, like those that you see here. Why do you need a grid? Well, for two reasons, basically. The first reason is that we want to represent shapes in a way that computers can understand. And the second reason is that we want to project an infinite dimensional problem down to a finite dimensional one. I mean, PDEs are in infinite dimensions, typically. Their solution is expressed in terms of functions that belong to, say, Banach or Hilbert spaces, which have an infinite dimension. Now, if you want to use computer to solve a problem, we absolutely need to project this problem down to upon subspaces of finite dimension. And to identify and to characterize spaces of finite dimensions, we need to use elementary functions, like, for instance, piecewise polynomial functions. And we have then polynomials on every elementary geometry here. So on every tetrahedra or on every ixahedra. How many do we need? Well, it depends on the accuracy that you want to recover. If you want a very accurate simulation at the end, we need very many elements. Like in this case, we're using here 60 million elements. Of course, the more elements you have, the most demanding the computation will be in terms of resources, in particular CPU time. The trade-off between accuracy and computational efficiency is always present when we go to the numerical simulation. So this is, again, kind of pre-processing. Now we have all what we need to start with our model. And then we have to move and try to represent the actual function of the heart, all the, physical processes that take place in the heart in terms of equations. And I'd like to, first of all, to give you an idea of what is going to be at the very end the model. The model will be able, hopefully, to express all the processes that take place in the heart. So the first one is the so-called electrophysiology. We want to describe the way the electrical field propagates through the heart, the way it impacts the ionic activity at the cell level, and then we need to take care of the mechanical activation. So thanks to this electrical potential, every single cell, the cardiomyocytes, will be electrically activated. There will be a force, which is called an active force, that will be generated at the microscopic level. Now, the aggregation of all these forces, the interaction between all these forces at all the cardiomyocytes level will eventually end up with a complete deformation of the muscle of the heart. And this will then give rise to what we call active and passive mechanics. I'll be more precise later on. Then we have another process that takes place, and this is the fluid dynamics in the four chambers, because we have blood, which is, of course, stimulated by the fact that the four chambers contract and relax. Then we have the dynamics of the valves. Then we have the perfusion, which is the process by which the coronaries, which are sitting on the surface, say, of the myocardium, will perfuse the myocardium, will bring blood to the myocardium, will nurture the myocardium. So this is somehow the synthetic view of all what is happening at every single chamber, for instance, the left ventricle. Then we have to reproduce it somehow for the other three chambers and to connect the whole thing with the external circulation, with the systemic circulation and the pulmonary circulation. So this is what we call an integrated heart model. So ideally, we should be able to do to set up equations for all these processes and linking all them together. We'll call each one of these processes a core model. So we need a core model to express equations, to formulate equations for each one of these physical processes. So we start with the very first one, which is the electrophysiology. By electrophysiology, we mean here the process by which the electrical field is generated and it propagates through the complete myocardium. Of course, we'll have no time to derive the equations. I want to show you the type of equation that you have to deal with. So the equation that are standing at the base of the electrophysiology are those equations here. They are called monodomain equation. This is the simplest version. We have a more complex version, which is called the bidomain, which is pretty similar. We'll have two equations instead of one in this case. So the variable is the transmembrane potential. So the potential, the difference of potential that you have across the membrane of every single cell. We have, as you see, the dynamic term. We have a diffusive term, which D, which is the conductivity tensor. And of course, it depends on the physical structure of the myocardium. Then we have the ion term. This one is expressing the reaction terms that occur at the cell level, at every single level. And it depends on two variables, U, which is the transmembrane potential, and W, which is the set of variables that describe the ionic activity at the cell level. And then they comprise basically the concentration of the different ion species, like calcium and potassium, for instance, but also some gating variables that are equal to 0 if a specific channel, membrane channel, is closed, and 1 if it is open. So it's a binary variable. And here on the right, you have the external stimuli that is induced by the sinoatrial node, say. So this is, mathematically speaking, a nonlinear partial differential equation, time-dependent, of reaction diffusion type, which is supplemented by a set of system of ordinary differential equations that are there to express the dynamics, to recover the dynamics of the ion species at the cell levels. Now, this is the action potential curve that I have shown you before. You start with a quick depolarization. This is followed by a short repolarization, by a plateau, and then a repolarization, and then back to the resting potential. And the whole process here takes place, as you see, in about 400 milliseconds. So less than half of a second, say. So this is the first set of equations. Of course, the ionic model may take many different forms. And here you see the names that are associated with some of those forms. Some of these models are completely, I would say, heuristic, experimental, based on phenomenological observation, on fitting curves that you can deduce from experimental evidence. Others are models that are derived in a, say, more systematic way. Now, if you solve this model numerically, you can indeed compute the propagation of the electric potential on the complete heart. You see here two phases of the same simulation. On the right, you see the propagation from the stimuli from the sinoatrial node before to the atria, and then to the complete to the two ventricles. Say you see them on a longitudinal cross section, while on the left-hand side you see the same simulation, but now at the level of, say, the pericardium, which is the external membrane of the heart. You see here the millivolt. For the ventricle, you typically go from -80 up to 40, and from the atria from -80 to 20. So here is the same simulation, but this is a picture, the one that you have on the left-hand side, that is better appreciated by the doctors, is more synthetic, but you see the activation map. So you have the isolines of the time at which single points are activated. So what you see here is a sinus rhythm, so it's a physiological beating of the heart, and you see the different times. You see here on this map, you see the different times at which the different say zones of the heart are activated. And on the right-hand side, it is the same simulation, but now you see the propagating front of the electrical field. It separates zones that have been already activated from those on which the electrical activation has not yet arrived. Of course, you can use it then also in clinical problems, say, the one that you see here for ventricular arrhythmias, for instance. So the idea would be to find the biomarkers that characterize the malfunctioning of a ventricle that is affected by very severe arrhythmia, like line of slow conductions, lines of blocks, and possible targets of ablation. This, in the end, is our goal, to try to help by means of the simulation that we see on the left, which are non-invasive, trying to help doctors to find the best possible spot, best possible points where to ablate. Ablate means basically to apply high energy, say high temperature, and burning, say, the tissue in those specific points, which are assumed to be responsible for the recirculation of the electrical field. So you already see here a possible implication or a possible use of the model that you have seen before, you can use them to better understand the physiology, even at more quantitative level, but also to try to help doctors to better, operate people who necessitate to be operated because of heart malfunctioning in a non-invasive way, and of course, in a more practical and economical way. But also you can simulate different type of scenarios before actually going to the operation and try to a priori try to guess which is the best possible result that you can achieve. The electrophysiology is the very beginning of the whole process, and then you have the cardiac, what is called active and passive mechanics. By that we mean the active deformation, the active force that is generated at the level of every single cardiomyocyte due to the electrical stimuli, and then the deformation of the whole muscle, of the whole myocardium, which is, of course, due to the fact that the myocardium is a solid structure which undergoes a passive mechanics behavior, namely when you solicit it, it will deform, but also it undergoes an active mechanics behavior, which is due to the fact that you have an electrical field which is applied at every single cardiomyocyte. So you have an equation or a set of equations that describe, say, the deformation of every single cardiomyocyte, and this is a nonlinear function. You see is an ordinary differential equation with a nonlinear term on the right hand side that depends on the elongation, say, on the concentration of the ionic species, and more particularly to the calcium ion, say. Then you have the standard equation of elastodynamics for the passive mechanics. You have the Piola-Kirchhoff tensor here, which depends on the deformation, d, and on the active force that is generated at this level here. And you have non-conventional boundary conditions that have to take into consideration the fact that the system is not isolated. You are just cutting it for numerical purposes for the time being, but you have to be careful on the way you prescribe boundary conditions on the upper surface, which is a virtual surface, and on the external surface, which is partly is a physical surface, the epicardium, but also partly is not isolated because it is in connection with the septum and with the rest of the lower part of the heart, namely the right ventricle. So you see here a picture that you can get from the simulation once the system has been solved numerically, and you see at different time levels the deformation of the myocardium. We go here from 0 up to 1.2 centimeters. So this is the first. There's the second core model, the one that has to see with the mechanical deformation of the ventricle. Then you have to couple the two things together. So you have to couple the active and passive mechanics with electrical potential propagation, with the ionic activity, and the mechanical activation at every single cell level or cardiomyocyte, say. So this is what we call the electromechanics core model. Of course, as everywhere in this presentation, we deal with what are typically called multi-physics problem. Multi-physics because you are coupling different type of physical processes together, and is also a multi-scale problem because you have different type of behaviors in space and time. You have different scales in space, different scales in time, and this make the numerical computation especially complicated. This is the coupled system, the coupled electromechanical model, where you see all the equations together, and with that you can actually reconstruct or simulate the deformation of every single ventricle. You see here the ventricle, the left ventricle, and but then you can also use it for having the deformation of the two ventricles. You call it biventricle, say, the left and right ventricle. You see it on the left-hand side. The R's that you see are indeed the fibers, the cardiomyofibers. I do not have the time to enter into this discussion, but cardiomyofibers are typically not available from data. So you have somehow to guess them, to reconstruct them from very incomplete set of data, and with this aim, we have to use extra partial differential equations to try to create, first of all, a potential, and then generating the direction, the local direction of the cardiomyofibers. On the right, what you see is the calcium concentration. So the different colors refer to the calcium concentration. So now we have an electromechanical model. You can go to the fluid dynamics. And the fluid dynamics is basically governed by the Navier-Stokes equations. We are making some simplifications here. We are assuming that the blood is a fluid, which is, as you know, not really the case, because it is a suspension of blood particles, right? Red blood cells, platelets, and white blood cells and leukocytes, say, on a bed, which is the plasma. But macroscopically, in large vessels and in large chambers, you can assume that it is indeed a fluid. We use a Newtonian representation of this fluid. We are assuming that it is incompressible and viscous. So you can use the Navier-Stokes equations. So Navier-Stokes equations are equations that translate the principles of mass conservation and momentum conservation, which are two physical principles. And they are expressed in terms of two variables. One is u, which denotes here the velocity field in the three space dimension. Of course, it depends on space and time. And the other one is p, which is the pressure, which is actually a Lagrange multiplier for the constraint which is given by this equation. This equation is divergence of u equal to 0. It expresses the continuity. It's called the equation of continuity. It is expressing the conservation of mass. So you have the continuity equation for the conservation of mass, and then these equations, which are vector equations for the conservation of linear momentum. This is the classical form of Navier-Stokes equations, with a little caveat, a little difference, which is due to the presence of this extra term that we'll not have in the standard Eulerian representation of Navier-Stokes equations. This extra term is there because we are actually trying to combine the Eulerian representation of fluid equations with the Lagrange representation of the mechanical deformation, those that you have seen before. So this hybrid representation demands for a little compromise, and the compromise is achieved if you introduce an extra variable. The extra variable is the rate of deformation of the volume occupied by the fluid. The fluid occupies a volume that changes in time because of the contraction and the relaxation of the ventricle. We want to reconstruct at every time instant the actual shape of this volume and the position of every point interior to the volume. And we want to compute in particular the rate of deformation, or rate of variation of these changes that we plug here to modify the convective velocity in the standard Navier-Stokes equations. So this is an extra variable which requires an extra equation. And you have different ways to recover this equation. This approach is called the ALE, arbitrary Lagrangian Eulerian. Lagrangian Eulerian because it mixes up the Lagrangian framework of the elasticity for the mechanical deformation and the Eulerian framework of the fluid dynamics for blood dynamics inside the chambers. And it's called A, like arbitrary, because you have many different ways to recover this velocity, because you have many different ways to go from the Dirichlet data, the boundary of the ventricle, through the lumen, the variable that describes the position of every single point at the interior of the lumen. Here we took the most classical, but also the easiest way, which is harmonic extension. We are just extending harmonically the position of the, endocardium to the internal volume. And we solve then a Laplace equation for that. So those are the Navier-Stokes equations that are in principle capable of producing the fluid dynamics, of simulating the fluid dynamics within the different chambers. And to give you a little evidence, let me start with the atrium. This is the left atrium, which is, remember, in the upper part of the heart. We see two different pictures, two different movies here. On the left hand, you see the a volume rendering of the velocity magnitude. We go from 0 to 70 centimeter per second. So quite a faster dynamic, say. And what we notice is that we have the 4 vessels here that are the pulmonary veins that are bringing oxygenated blood from the lungs to the heart. And we have 4 high-speed jets from the pulmonary veins, and they are mostly impacting during the systolic filling. The systolic filling is the one when the ventricle will contract. The ventricle will contract. On the right-hand side, you can get an idea of the coherent structures, vortical structures that are generated during a heartbeat. We are here, we are, presenting the vorticity of the blood according to the Q criteria. Q is a variable, is 1/2, as you see here, the rotation tensor squared minus the strain rate tensor squared. So what we can notice here is that we have 4 vortex rings that are generated from pulmonary veins. This is called the energy production phase. And after impacting with the jets, they form coherent structures that become smaller and smaller until they disappear. And this is what is called the energy dissipation phase. Now, the Reynolds regime that characterizes the stability or instability behavior of blood flow is changing continuously over the heartbeat. Roughly speaking, we can say that in a physiological heartbeat, in a sinus rhythm profile, the flow is does not become turbulent. There is no time and no space, not enough time and not enough space to develop a fully developed to reach a fully developed turbulent flow. The flow is somehow permanently in a transition phase towards turbulence without reaching the turbulence, however. Now, you have a different perspective here when you see the complete left heart. You see then the atrium and the ventricle, and we see 3 different phases. What you see on the upper part is numerical simulation. On the lower part is 4D flow MRI, what is called 4D flow MRI. 4D means 3-dimensional in space, and we have an additional dimension in time. And you see here 2 different phases. Always reproduced is the velocity field. The modulus of the velocity field is just after the mitral valve opens. The mitral valve connects the upper part with the lower part of the heart, so atrium with the ventricle. This is before the mitral valve will close, and this is when the aortic valve opens and blood is ejected into the ascending aorta, say. We have the valves. We have the valves, and this requires an extra type of modeling. And here, there are plenty of possible models that you can employ. You can go from, the simplest one to the most complex ones. The most complex ones are those that treat the valves as 3-dimensional objects. So we need nonlinear mechanical models for them. What we see here is a kind of intermediate approach between the most complex approach and the simplest one, which is simply based on reconstructing the shape of the valves during the heartbeat. So this consists in injecting a modification in the momentum equation of Navier-Stokes equation, say. We have this extra term, which is a kind of extra source term, and it depends on several quantities. First of all, gamma K is the actual surface of the valves. What you see here, gamma 2 is the aortic valve. Gamma 1 is the mitral valve. RK is a coefficient that, expresses the resistance that the valve opposes to the fluid flow. Epsilon K is the thickness, actually the half thickness of the leaflet of the valve. delta gamma k, epsilon k is a direct delta function. So basically what we are doing here is to add an extra term, which is only living on the valve surfaces. And this extra term depends on the difference of the velocities between the actual velocity of the fluid and the velocity of the formation of the valves, say. So it's a kind of direct source that we are adding to the momentum equations of Navier-Stokes equations. And with that, we can now simulate the fluid flow across valves, past the valves. What we see here is the aortic valve, which is a 3-leaflet valve, which opens and closes. When it opens, it allows blood to be ejected from the ventricle into the ascending aorta. And when it closes, it prevents a backflow from the ascending aorta, of course. Now, this is another simulation of the same kind. As you see, different representations here. On the left, you see the displacement. We are considering now the left atrium, the left ventricle, and the ascending aorta. In the middle, we see the vorticity that is generated when the mitral valve opens. High-speed jets are coming from the left atrium and are filling the left ventricle. They produce the formation of O-ring. Maybe you can see also this one. Yeah, okay, of what are called the O-ring, O because they have a form of an O, which are coherent structures that are rolling through the leaflets of the mitral valve. Then this vortex breaks into much smaller vortices with much smaller coherent structures that are filling the left ventricle and moving toward the apex, as you see here. They move toward the apex, which is the lowest part of the left ventricle. And when the systole begins, when the left ventricle contracts, those are marked by the opening of the aortic valve, this one. Then the structures are flushed out into the aorta, and they then dissipate. And at the same time, new jets are entering in the left atrium, but they're weaker with respect to the ones that we have observed in the diastolic regime. So we can get a complete reconstruction of the flow field here, which is fully characterized by the displacement dynamics, the vorticity dynamics, and of course, the velocity field. These are detailed, what happens in the ascending aorta. That's a very critical point because many pathologists get their origin just in this very point. And what you can observe here is that in the systolic phase, which is the one when this valve, which is the aortic valve, opens, and you have a jet that is ejected from the left ventricle into the ascending aorta. You see a high-speed jet. You see that the jet impinges on the wall of the ascending aorta, on this wall here, and this yields a recirculation region downstream. Here you have recirculation, and downstream the jet, and you have very high wall shear stresses. Now the wall shear stress is basically the tangential forces that operate on the surface of the wall, and their dynamics is particularly important because the oscillations of wall shear stresses are very often the basis of the formation of plaque, of atherosclerotic plaques, which are, I mean, pathological evolution, pathological, say, the formation of the vessel wall. Now, what you have seen so far is a model, an electromechanical fluid model that concerns the heart, or more specifically the left heart, what you have seen so far. Now, the point is that the left heart is not in the middle of nowhere. It should be connected with the rest of the circulation. And as you can understand, since you have hundreds of large arteries and billions of capillaries or small arteries on small vessels, say, in the circulation, there is no hope to use three-dimensional models to catch the whole dynamics. So this would be out of reach from the numerical viewpoint, from the computational viewpoint, say. So we need to simplify the model. And one way to simplify it is to use what we call zero-dimensional models. Zero-dimensional models are models that are based on the analogy with electrical circuits. We call them 0D because the variables only depend on time and not on space any longer. So the space dimension is no space dimension. We only compute average quantities. So you have to build up an electrical circuit, or say a series of electrical circuits that are capable of reproducing the complete circulation outside the heart, say, and then to connect it with the three-dimensional model for the heart. And of course, a very critical issue here is how you connect 0D models with three-dimensional models without losing too many physical information. This is the result that we obtain when we connect it with the external circulation. Again, is a three-dimensional analysis for the blood flow and the contraction in the left heart, and you don't see it, but this is connected with the zero-dimensional circulation on the rest of the body. Now, there is a last point that I would like to address, and it's the coupling between the deformation of the structure and the fluid dynamics. And here we have to couple together the Navier-Stokes equations with the passive mechanics with the harmonic extension in order to recover the lumen of the heartbeat. So this is called the fluid structure interaction. And now we are done. What we still missing is the perfusion of the myocardium. If we want to inspect what is going on at that level, of course, this part of analysis is necessary when problems arise by the fact that the coronary arteries are not clean enough, are not open as they should be to perfuse the myocardium in the right way. Now, can we describe this fluid dynamics as well? Well, the difficulty here is due to the fact that we have several, say, order of 10 large arteries, large coronaries that are sitting on the external surface of the myocardium, but then they enter into the tissue and they bifurcate and bifurcate and bifurcate and give rise to a very, say, complex network of very small vessels. So we like to be able to catch all this system of large and medium size and small size and macroscopical size vessels. And for that, we are using a multi-scale geometrical model. We first start from the vessels of a real patient, those that you see here under my mouse. Those are the most important vessels that you can catch and you can reconstruct. You can see from medical images, but then we move from there and we want to reconstruct also the intramural vessels. And here we have to use mathematical artifacts, mathematical models to generate the intramural vessels because they are basically impossible to see with the standard clinical images. And from the modeling point of view, we are using three-dimensional Navier-Stokes equations for the most important vessels, the epicardial vessels, and we use a multi-compartment Darcy models for the small vessels. We use a Darcy model because we are simulating this flow dynamics to the one that you see that you have in porous media, but use a multi-compartment because you want to distinguish between vessels of different size. And we use at every point 3 different compartments. So they're not separated spatially, they're coexisting at every single point, but they are expressed by different, say, permeability and porosity coefficients that we have to simulate and we have to reconstruct by, say, parameter identification. So we have further system of equations. Again, the Navier-Stokes equations in the large vessels. Then you have the multi-compartment Darcy laws, and then you have interface coupling conditions. Again, it's a bit too involved to be discussed in detail, but for those of you who are interested, of course, I can send material, I can send papers. So by this system, we can help doctors simulating the perfusion of the left ventricle, for instance. And we did this exercise to see the way you can avoid having a patient undergoing the ciclet or say an effort to see the way his muscle is perfused. And so we do it by numerical simulation, and then we compare. We have 9 different patients, and what you see is that we basically have a good reproduction of the perfusion mechanism. The errors are below 1% on 9 different patients. So it's a preliminary investigation, but it's somehow interesting. So let me conclude by saying just a few words on the numerical aspects. I completely disregarded the numerical approximation, but of course, to get the results, we need to go to the numerical level. And here are the most important issues. We have multiple scales, as I was saying before. We go to nanometers to centimeters to the organ level in space. We go to microseconds, which is the characteristic time at which the force is generated at the cardiomyocyte level, to seconds, which is, say, characteristic time of a single heartbeat. We have lack of data. That's very important. We don't have initial conditions and boundary conditions, and we lack many coefficients. So we need to work a lot in terms of reproducing those data that are missing. This is one very important point that is behind the numerical solution of these partial differential equations. We have geometrical nonconformity. We generate grades with different methods from, say, the fluid dynamics, from the muscle, from the myocardium, and we have to use patching algorithms to make sure that the different grids communicate in the right way. We need splitting algorithms because you have this complete model that is a gigantic mathematical model that you cannot solve all together at the same time in a single shot. So you have to split all these different core components, but then we introduce splitting errors that you have to understand and to dominate and to estimate. We need scalable preconditions because we need parallel computers and we need to solve all this in parallel. So you have to make sure that your algorithm scale properly when you go from, say, 10 CPUs to 1,000 CPUs. We need model order reduction because the simulation of a single heartbeat takes order of days on one of the biggest supercomputers in Europe. And if we want to interact with doctors, we have to take this down to the level of minutes. So you need model order reduction. And of course, you have to account for the variability and uncertainty. So this is all what I wanted to say. I had a final subject that I will not be able to touch, and it was, say, the application to this to the COVID-19 contagion, and try to see the way we could adjust these models to the COVID-19. But I think I used all my time, so I'll prefer to stop here and, of course, answer your questions in case you have questions. Thank you very much.
So thank you very much, Professor Quattroni. On behalf of everyone, thank you very much for your very instructive and interesting talk. So we have one question straight away from Nick Hill. The inner wall of the ventricles is not smooth. How important do you think this is, and how could this be modeled?
The inner wall of the ventricle, meaning the endocardium, I guess is endocardium, right? I guess you mean the endocardium.
Yes, that's what I mean, yes.
Okay, you say that it's not smooth, right? Or it's not thin?
Yes.
It's not thin or it's not smooth, sorry?
Smooth.
Oh, it's not smooth, all right, okay. Well, I mean, we try, I guess I don't have a picture here that would illustrate the way we simulate it. We try to go with our grid to catch this detail. Obviously, there are plenty. We have to take into consideration the chordae tendineae and the papillary muscle. We have them in our grid, and so at some extent we are able to go at that level, say, of I guess this is what you mean, right? Yes. The non-smoothness is due to the fact that you have this very tiny structure. So we try to account for them, at least for the mechanical model. We are not yet there for the fluid dynamics model because this would require far more many points grid points, and it will be very difficult to go at level of detail with the fluid dynamics. But you try to reproduce them at in our mechanical model.
Thank you.
Thank you. Is there any other questions?
I could ask another one.
Please.
Okay, go ahead, Dick.
So you're using a monodomain model for the electrophysiology. Do you think using a more complicated model would significantly change your results?
I actually here I actually presented the monodomain, but actually you also use the bidomain model. So I would say that in a sinus rhythm in healthy condition, the monodomain equation is, according to our experience, accurate enough. In some circumstances, you may be it might be wiser to use the bidomain equation. We actually have both approaches, and sometimes you even use an even easier model, which is based on the iconal equations. So it's really depend on what you want to observe. So we can tune the complexity of the model depending upon the type of answer that you want to provide. And I would say that this is a kind of general attitude that we have. Of course, as you can easily understand, this monster model is in principle capable of reproducing the complete heart function, but is also tremendously complex from the numerical viewpoint. And most often you do not need it in all its complexity, own complexity. So depending upon the type of interaction that we have with the doctors, medical doctors, we sacrifice some of these components and we highlight only some subcomponents, what we call core models. For instance, for the arrhythmologist, the fluid dynamics in general does not play an important role. So we completely segregate it. But when, of course, we want to provide answer to people interested in replacing a valve, like the aortic valve or the mitral valve, then what counts is what most count is the mechanical deformation and the fluid dynamics. And in that case, we completely neglect the electrophysiology component. So as you see, we can tune the complexity or the easiness of our models depending upon the type of observation that you want to make. But most often we use the monodomain equation, even though we have the bidomain equations.
Thank you.
Welcome.
There are a number of comments in the chat saying how much you enjoyed the lecture. Any other questions? Yes, there's a question coming up in the chat. I don't know if you can see the chat, or do you want me to read the question out?
Sorry, I don't see the chat. I don't know why.
One can observe an increasing number of publication titles mentioning advanced statistical, in particular machine learning methods in applications to mechanical problems. Do you see any relevance or successful use of these methods in multiphysics of the heart?
Very nice, very interesting question. Well, actually, I focus my presentation only on the modeling side, which I would say using first principles, physical principles to derive equations. As a matter of fact, we have many different ways to combine this with machine learning. And we basically do at multiple levels, but let me mention a couple of them. One is the way these models, those based on physical principles, can supply information to machine learning algorithms. For instance, in cases where you have paucity of data, not very many data, we can generate training set or enrich the training set of machine learning algorithms or neural networks algorithms by producing synthetic solutions obtained by numerical simulation. This is the case where, for instance, in problems which have to see with the pathologies of the heart rhythm, you do not have enough patients, not enough data generated by patients. So we produce synthetic solutions by our models to enrich the training set for neural network-based algorithms. That's one way. So go from physical models to machine learning algorithms. And another way is how to use machine learning algorithms to solve the inverse problem. You have several inverse problems here potentially, namely the problem of identifying coefficients that are not available from clinical data. So using neural networks to solve inverse problems to have a better idea of a better quantification of of coefficients that characterize our physical models that are not available from data. And another possible approach is that you see, I mentioned before the fact that we have here multi-scale models. We go from the macro scale, the one that you see here, to the micro scale, the one that you see at the cell levels of the cardiomyocyte level. So, and there typically you need constitutive equations. And most often you do not have constitutive equations. You do not have equations, you do not have basic physical principles that describe behavior at the microscopic level. And here again is where artificial neural networks can help to guess dependence of, say, different variables at the microscopic level. Here are three examples where, but I could quote many others. Well, let me at least quote a fourth one. I mentioned before the necessity of generating model order reduction algorithms. Let me see if I have it here, right? So model order reduction. By model order reduction, I mean the way you start from the PDEs and you project them onto a space which much smaller dimension, which is obtained by linear combination of snapshots that are obtained by statistical principles, say. So there are many different ways to generate model order reduction algorithms or problems. Some of them are purely based on physics-based models, like the ones that you have seen here. Others are based on machine learning algorithms, and they're very effective in general. So that's a fourth way where you can actually take advantage of combining the two approaches together. And my personal opinion is that this is just the beginning. Now, PINN methods introduced by George Karniadakis and colleagues is another way where we start from machine learning algorithms, neural networks, and then we compute in a symbolic way residuals that are based on differential equations, and we use them in the loss function to improve your neural network. So I see many different interplays between the two approaches, and I guess this is the way to go.
Thank you. I think we've got time for one last question. Firstly, congratulations on your really great presentation. So the question is, for which kind of heart diseases do you think that the 0D circulation approximation should be relaxed? Or is it working well for a large variety of patients and diseases?
Well, first of all, I think the 0D are, I would say that they are somehow essential if you really want to catch the dynamics of the broad scale, or the whole circulation scale. You cannot avoid them. Of course, it depends on what you want to observe. If you want to observe, for instance, diseases that originate at the level of capillaries because of the deformation, pathological deformation of red blood cells, like in malaria, for instance, then in that case, you have to go and see with very sophisticated methods what's going on there. But if you want to see the effect that the global circulation has on the heart, for instance, then you really have to go and use zero-dimensional models or reduced order models at that level. So zero-dimensional models provide information of variation of average quantities, the variation in time of average quantities in space. So they are very good in reproducing, for instance, pressure wave analysis, or the flow rate that you have on the valves, if you want to use, say, an external method based on zero-dimensional models, or macroscopic quantities. So there are many different circumstances where you can actually live with zero-dimensional models. And actually, as you might know, bioengineers love this type of approach because you can catch many information, quantitative information at that macroscopic level. And I guess this is also important to inform the more sophisticated 3D models a priori, to have an idea what's going on, what you should expect. And then, of course, you can refine your analysis with three-dimensional models. So there's plenty of room as 0D models, and they can live by themselves if in order to provide specific answers. But otherwise, if you need more sophisticated and more precise and more accurate responses, then you actually need to go to use three-dimensional models. But in that case, zero-dimensional models are very important to provide a feedback mechanism or to get a closed loop formulation.
Okay, thank you very much for answering all those questions. I think I'll draw this seminar to an end. I'd like to thank everybody for coming, and I'd like to thank Professor Quattronioni again. And really sorry that we can't meet you in person now.
Thank you very much. Thank you for your patience. Thank you for inviting me.
Thank you.
See you.
Bye-bye.
Thank you.
Bye. Bye to everyone. Bye-bye. Thank you.
Thank you very much, Alfio. Yes, it was great talk.
Very welcome.
And it was Raimondo who asked the last question.
Sorry?
And it was Raimondo who asked the last question.
Oh, okay.